extension of the first video that we did. Whereas your first video was a cursory overview of basic information about cellular energy transformations that you'll need to know. This is going to go slightly more in depth on cellular aspiration, and it's required for honors classes only. So your two objectives that you have here, one is that we're going to be talking about cellular aspiration. So the first thing that you need to be able to do is diagram and talk about the parts of the mitochondria. So up until this point, you just had to do basic labeling inside cells. Now we're actually going to talk about what those structures are inside the mitochondria. And the second thing you're going to have to do is take a look at the 3 steps that occur in aerobic cellular aspiration. You're going to have to be able to provide their locations, a general overview of what occurs in each of these processes, and what the product is. So quick clarification statement. What we have here, this is a top-down flowchart of what actually goes on in cellular aspiration. Now, you'll notice, just first looks right here, that it is a lot more complicated than just oxygen and glucose goes in and carbon dioxide and water and ATP comes out. There are a lot of steps in here. We are going to go over some of the 3 basic steps that you find. So glycolysis is our first step. The next step we're going to be talking about is the Krebs cycle, which you'll notice if you follow kind of the line, your flow chart down the right-hand side, you see Krebs cycle. And then the last one we'll be talking about is electron transport chain. What I want you guys to notice about the chart over here is that after glycolysis is going to produce something, 3 carbon chains that are called pyruvates or pyruvic acid. And you'll notice that it splits here. And to one side we have O2 not present, and the other side we have O2 present. What we're going to talk about today and what you're responsible for is going to be the aspect of cellular respiration that is aerobic cellular respiration. So that is, aerobic means requiring oxygen, so occurring in the presence of oxygen. There is also another part of cellular respiration that can occur in an anaerobic environment. So this does not require oxygen to take place. Now, we still do anaerobic cellular respiration inside of our cells, and if you're an athlete, you probably have felt the byproducts of anaerobic cellular respiration. If you run for long periods of time, once your body, your muscle cells cannot get enough oxygen from your respiratory system to create ATP using aerobic cellular respiration, your cells will convert to using anaerobic. And if you kind of follow this flow chart right here, what you notice is that, so we get to pyruvates, your pyruvic acids produced from glycolysis, and we notice that fermentation. Fermentation is the process that's going to produce alcohol, and ethanol is produced by some bacteria, and this is how alcoholic beverages. But inside of our body, what fermentation produces is something called lactic acid. If you've ever run or done any sport, lactic acid can cause some pains and cramps inside your muscles if you allow it to build up. So you do have anaerobic or non-requiring oxygen cellular respiration that occurs inside your cells. Today we're just going to talk about aerobic, so we're going to focus on that right-hand side. So here's our mitochondria structure. There's 4 basic things you need to know. The first 2 are simple. So we know that mitochondria have a double membrane. Your outer membrane is going to be oval-shaped, and that'll form sort of that basic shape that you see of your mitochondria. Your inner membrane, which is labeled number 1 on here, if you'll notice, it ripples and folds. So it's still one continuous membrane, but what you're going to find in here is that it is grooved. So it's got lots of bends in it. Each of those bends is called a cristae. So the cristae itself is the folds in the inner membrane. So those folds, they have their own name because parts of cellular respiration are going to occur at the cristae, so at those folds. And then the last 4th thing is the matrix. So the matrix refers to the inner fluid. So remember, this isn't air inside of here that's filling up these empty spaces between your membranes. It's a fluid, a cytosol or cytoplasm-like fluid that fills up the inside of the cell. So we have an intermembrane fluid that would be between our outer and inner membrane. The matrix is going to be inside the inner membrane, so that orange colored stuff. So here are the 3 parts of cellular respiration. Now in your notes, what I've done is given you a copy of this chart. What we're going to do now is fill in the first 2 columns. So we're going to talk about ATP produced. Again, this is going to be under ideal conditions. So when I give you these numbers of how many ATP are made during each part of cellular respiration, I'm really giving you best case scenario. So the 3 parts of cellular respiration, the first one is glycolysis. The second part is called the Krebs cycle or the citric acid cycle. Those are 2 terms to describe the exact same process. Krebs is named after the person that discovered it. Citric acid refers to the carbon molecule that enters into the cycle. And then the third thing is the electron transport chain. So if we start with glycolysis, and we'll go into these a little more in depth later. Glycolysis, the first part of cellular respiration, is going to produce 2 ATP under ideal conditions, and this is going to take place in the cytoplasm of the cell. So you know how we said that all bacteria, all organisms do cellular respiration? Glycolysis. Glycolysis is one part of cellular respiration that all organisms do. So even bacteria that don't have a mitochondria, they can break apart sugars inside their cytoplasm. They can do glycolysis. They can make some ATP in their cytoplasm. And again, you'll notice that that last column has stars in it, and that's because we're going to go into these more in depth in later slides. The Krebs or citric acid cycle, so the product of glycolysis is then going to enter into the Krebs or citric acid cycle. It's going to get broken down even further, and you're going to produce 2 ATP in ideal condition from the Krebs cycle. This is going to take place in the mitochondrial matrix. So this is going to take place in that fluid inside the inner membrane of your mitochondria. The last one, this is your big ATP producer. So this is where all of that energy comes in. This is what makes it worth it to have mitochondria inside your cells. Electron transport chain is going to can produce 32 ATP in ideal conditions. And this is going to occur in the mitochondria as well, but across the cristae. So across those folds in the inner membrane. All right, so big hint on these next 3 slides. Anything written in green is really the key information you want to have in your chart. I'll expand upon things in there as well, but you need to make sure you have the green information in your chart. Please feel free to stop as much as you would like. So here's a diagram of what goes on. So if you take a look, our blue box right here that's circled in red, this is where we're starting in glycolysis. Now you'll notice right here that it says in black underneath it cytosol. Cytosol is just another name for the cytoplasm, so for that water-based substance that fills up your cell. So here's glycolysis occurring outside of the mitochondria. Now glycolysis, if you look at it, it looks like glyco and then lysis. Now we talked about hydrolysis, where you can split apart organic molecules using water, and we said that lysis means to break apart. So glycolysis is literally the breaking apart of glucose. So here's your green part you need to write. So 6-carbon glucose. We know that glucose, the formula for it is C6H12O6, meaning there are 6 carbons. What's going to happen in the cytoplasm is it's going to be split into 2 3-carbon molecules called pyruvate. When you split apart those molecules, and here's your big idea in red, when you form bonds, what you've essentially done is stored energy in those bonds for later use. So by breaking those bonds between those carbons and glucose, what the cell is essentially doing is releasing energy. And it's going to use that energy that it releases by breaking apart those carbons to create 2 ATP, so create 2 energy molecules right out there in the cytoplasm. You'll also notice, so the arrow that comes down from your cytoplasm shows you that ATP that's produced. You'll notice there's another arrow that comes across the top, and it says electrons carried via NADH. Now, in addition to using energy from breaking apart these carbon molecules to make ATP, the cell is also going to store a little bit of energy in a high energy hydrogen. So if you notice right here, NAD, that H that's on the end right there, that is a hydrogen that is carrying a lot of energy in it. So NADH, think of that as kind of being like an energy savings bond. So this is going to be used later in the electron transport chain, which is where this diagram shows that NADH going, to create those 32 ATP, so that real powerhouse part of our cellular respiration. So again, makes NADH, and I put the H in red because remember that's where that energy is getting stored, because NAD is a regular molecule, floats around inside the cytoplasm, and then when a high energy hydrogen becomes available, it's going to snap it up, and it's going to carry it to the electron transport chain where it'll be used to make more ATP. The next part of cellular respiration. Okay, so we broke apart glucose, we made pyruvates, so we made smaller carbon molecules, because we started out with a 6-carbon molecule glucose, now we're down to 3. Those smaller molecules move into the Krebs cycle. They're going to break down before they enter into the Krebs cycle, so instead of having a 3-carbon pyruvate, we're going to end up with 2-carbon molecules going into your Krebs cycle, and those are called citric acids, which is why it's sometimes called the citric acid cycle. What's going to happen is that these carbon chains are going to be broken apart even more to make 2 ATP and make more NADH and FADH.